Hi,
Hi, how are you?
I'm good. How are you?
Good. All right. Um, well, are you ready to give an update on your week's
Yeah,
progress? All
just
right.
give me a minute. Yeah, should we wait till someone else joins in? Or I can
No, we
start.
can go ahead and get started.
Okay. So, coming back to what I've been doing, Sustain Hub. This is open the full project that I'm working on. So in week 1 and week 2, this is what I had promised in the timeline that I will implement the baseline and the environment. Out of which I did half of it last week in week 1. I continued doing that in week 2. And I also started with the multi-app thing, and it's almost done. It's not perfected yet, but it's working. So this is the file structure that I have right now. It's more or less the same as last time, and not many changes. Only that I added the multi-app bandit thing, the file. So basically, over here, what I've done is earlier, what was happening till last week was that the environment was set up, and as the tasks were being generated, it was being assigned to the agents randomly. So that time, only the environment was running with the tasks being allocated randomly. But now we actually have the multi-armed bandit, and in multi-armed bandit, it focuses on exploration and exploitation. So first, the agents will explore the tasks, and once we know which agent should perform which task, it will exploit it. So for exploration, we just have random. We use np.random so that the agent can actually explore, and then we use Thompson sampling or Bayesian whatever the Bayesian formula to actually have the exploitation part. And what happens in multi-arm, like what I've done in multi-arm bandit is that the multiple arms are actually the agents which will do the task allocation part. So that's what I did this week. And this is the output that we got. That earlier the tasks were being assigned to random agents, and then eventually they were communicating, and the correct tasks were being assigned to the correct agents. So there's no randomization in it. So that was achieved well. And yeah, then the same thing, like I updated my README file so that if anyone wants to go into the repository and run the whole environment along with how the multi-arm bandit part is working. So that was done. Yeah, I added the blog for week 2 as well, which is over here. And I'll send the link again in the chat box or the chat. Yeah, so this was week 1's blog, this is week 2's blog. And I'm not really sure whether I'm going on the correct path with my blog. This is my first time with blogs. But yeah, and so this is the timeline. Actually, multi-arm branded was supposed to be a part of week 3, but since I had time in this week, I actually went ahead and implemented it so that if I face any issues later, I do have a buffer to always cover up with my time. Yeah.
Okay. Well, yeah. Thank you. That was very good. Yeah. So that's good that you're kind of ahead of schedule. Be
Yeah.
aware that, like, yeah, you have the buffer. That's good, but also that if we get to a point where you run out of things to do, that you kind of think ahead
Mm.
and say what
Okay.
will need to be done after. And at the end of the summer, I want everyone, all three students, to think about what needs to be done later. So, we might create a project board towards the end of or we could create a project board now if you'd like, and we can put in issues that are, kind of longer term
Okay.
issues. And so, at the end of the summer, when the project is over, we'll have obviously some things completed, but then some
Mm.
things that are
Yeah.
going to be, for longer term maintenance or development.
Like, what do you want to include in the project board? Is it like the continuation of the project or
Well, it would,
what
yeah, it would be
exactly is it?
nice. And we've done this the last few years. It'd be nice if we had a
Okay.
project board for just kind of managing issues in the repository. So you have
Okay.
you've already got some things in the repository, and so just say like this is the thing that even if it's like something that you're going to do like next week, you could create an issue for it. You don't need
Okay.
to create issues for everything, but just things that maybe even to keep things straight in your own head to say, this
Right.
is an issue that maybe relates to what's in the repository, or
Right.
maybe something that is in another part of the project that you might think is, something that can be extended out to the other parts of the project.
Yeah. I remember seeing this in Sarah's projects actually. Like she created those issues, and then once she did it, okay, I got it. I got what you're saying.
Yeah, yeah. Jesse
Yeah, I got it.
was managing those last year, but we can set one up. I
Yeah.
think you have. Do you have administrative? I think I gave everyone permissions.
Yeah, I get it. Yeah,
Okay. Yeah.
I have it. Yeah.
All right. It's
Okay.
good. Oh, do you know why they call it the multi-arm bandit?
Because like multi arms, right? Like multiple arms,
Right, but what does it refer to?
like an octopus or something. Some animal, it's something like
No,
that.
it's because it's a slot machine. It's like
Oh yeah.
when they
Okay.
have
Oh
a
yeah.
slot
I
machine
get it.
arm and people pull it, and then like the idea is you have
Yeah.
multiple random processes that
Okay.
you pull and you get them to, yeah.
Yeah,
But that,
I've seen this example, like the 7771, the same is supposed to like I don't know how to put it, but I've seen that item. I think I put it in the very first week PPT where I presented.
yeah.
I got it. Yeah.
All right.
Yeah.
Yeah, I was glad to see that you had your GitHub repository ready and going here. So let's take a look at the organization on these repositories. I'm not saying that everyone needs to have everything up right now, but I just want to see. So this is Sustain Hub. This is what um, Vidhi was talking about. So she's got her like agents and tasks. So some main, some files that are kind of in there already. And that looks good. And then you have the README, which of course, we're not quite at the point where we're writing up the final report, of course, but we want to get that in place. So I want to, well, at the end of the uh, summer, everyone's going to have to submit their uh, projects, and usually through some link. They'll ask you for the link, and I'd like for people to host it in the repositories that we discussed, so that we can have like something laid out like this, where you have a README. The README is it's going to be kind of not well, it's going to be this sort of thing where you have the instructions, but also maybe like more information about the motivations and things like that. And you don't have to do it in one README, but like the idea is that when they look at your project, they can tell that there's been significant work done, and then they can also clone the repository and run it. And that's kind of the idea behind. Although um, they've expanded the criterion in recent years so that you don't actually have to produce a working program, but it's always good to have. I think for the projects we're doing this summer, to at least have some aspect of it that runs, or some completeness to it. So this is going to be kind of the way that we'll lay out all the repositories. So that's Sustain Hub, and then we have our DevoGraph repositories. I'm not sure where we are with that. We'll get an update from Lalith and maybe Jayadratha at some point to get like to see where they are with that. But this is, of course, the place where we'll host the DevoGraph repository, or actually there'll be directories underneath this repository. So if you can push things here, that would be great. And I sent again everyone an invite to the appropriate GitHub repository. Okay. Well, thank you, BT, for the update. Lalith, are you ready to give an update?
Hi Bradley, hi everyone. Since Lalith is not here, so I just you can. I was talking to Joy recently. I think he will be joining soon. He's stuck in rain right now somewhere, and he had his defense I think public presentation yesterday. So now he will be available to like contribute like very actively. Like till now he was like little occupied with the presentation and like some travel stuff.
All right.
Yeah. Lalith has also mentioned.
So he was defending a thesis, or
Yeah, I think thesis defense is still is still left. But yesterday was his public presentation. So we have like two steps. First is presentation, and the second is defense. It is, I think, next week or something like that.
all right.
So yeah, he'll be graduating soon, and I think he has plans to join PhD and like continue research in graphs.
Yeah. What was his topic for the thesis?
I am not quite yet sure. It is something in temporal graphs. I don't remember exactly what it is. I wasn't. And he also got accepted recently, like I think in some KDD conference, some workshop paper. Yeah, in temporal graphs, temporal graphical learning. So he just told me today. I think the news came like today or yesterday itself. So, yeah, I would love to hear more. Like if he can present things here, he'll be joining soon, like in a few minutes. He texted me just now.
Okay, that's good. Yeah, thank you. Yeah, thank you, Mehul. That's great. Did you have any updates on your own or of your own? Or?
No, Bradley. I'm actually occupied with something. I'm actually learning more on AI agents, like what Vidhi is doing. I would love to learn more from her.
Okay.
Yeah, so like, I'm actually very much occupied with that.
Oh,
Yeah.
well, that's fine. Yeah, that's good. Yeah, thanks for the update. Rohit, hello.
Hello, yes, sir.
How are you?
I'm good, sir.
So, what do you like? Give a short update about what you're up to these days, or what's going on?
Recently, I was just reading about the code base of DevoGraph, and somewhat I understood it, but I don't know how, what to contribute there and how to. I mean, I have read some of the docs, went to that C elegant website too, DevoWorm, went through the data sets, but I don't know exactly on which data set I should work and how to start work and how to make small contribution. Right now, like if I think of something, see, generation, hypergraph generation or something, then it's kind of a big task. So if I get some small data set on something, then I could start working or contributing. So yeah, this is the scene current.
Okay. Why don't we look over the repository and I'll discuss it a little bit. So yeah, this is the DevoGraph repository. So we have, this is kind of like a collection of the last 3 or 4 years of work. So, and there've been different projects, so it's hard to say kind of where things are in the current state. So I mean, I don't want to say like we're not at the point where we have like a formal program and a formal version and we're updating it. It's more like we have some approaches that we've kind of pushed to the repository, and we have, we had like an original sort of pipeline, and then we've kind of gone beyond that. So you'll see these folders: stage 1, stage 2, and stage 3. So the idea there was that we started off with this idea that you would take data like image data, microscopy image data specifically, and find features, apply some sort of machine vision algorithm or something, get features out of those images, and then work with those features to build graphs. So stage 1 was the image processing. Stage 2 was sort of the assembling graphs, and then kind of stage 3 was like doing this layer of analysis or topological data analysis. And this was the stuff that Pocky was working on last year. Stage 2 was, we had a couple people working on that. So we had Himanshu working on that, Himanshu Shogol. And then in stage 1, of course, we have this machine learning component, which is, of course, our DevoLearn software, and among other things. So, that's stage 1 is sort of complete. Stage 2 is, this is where we kind of went off into hypergraphs and temporal graphs and growing graph neural networks. And then stage 3 is this topological data analysis, where we have this, we use these mathematical tools to compute like higher order shapes and simplices of the networks. And so a simplex of the network is just simply sort of shapes that emerge in the network, like diamonds or crosses, or things like that. They're motifs that you can describe topologically. And so that's where we basically are with this repository. So things like growing GNN, that was from last year's DevoWorm, that was Pocky, and Jayadratha is also working on that this year. The idea that you can take data, we have data from our embryogenesis data from C. elegans. We have the cells, we have their position, and all that. And then we can put that into a network that allows you to model the growth of that process. So, we'll start with time 0, where you have one cell, and then you'll move to time 50 or time 100, and the cell divides into 2 cells and 4 cells and 8 cells and 16 cells. And then that forms some sort of spatial network where you have a distance between cells. That distance maybe represents chemical signaling, or it might just represent their location of their centers, their centroids. And then we build these networks growing out from the single cell case. So the idea is that these networks are growing, things are attaching to them, and they're growing in size. And then from there, we try to get to like an end state where the network is complete. So, there's a whole software approach to this. There are other approaches that might be promising, but Pocky was working off of some existing software. DevoGraph was what Mehul was working on last summer. So Mehul was working on this sort of DevoGraph approach, where you have nodes within hypernodes, edges within edges within hyperedges. So you have these hypernodes which contain a series of nodes. You have edges which contain a bundle of hyperedges or a bundle of edges that go from the nodes within the hypernodes to another hypernode with nodes. And so it's kind of like this nested graph. And Mehul worked on a lot of the sort of the basis for this approach, and that's in this repository. We have some data here, and this is where we have some of the data. So we have a CSV file here and a cell phenotype lineage data file here. And this is where it gets a little strange, because we don't have, I think we need to normalize some of these data sources, because I think we've gotten some data sets involved in, based on what people were interested in, and we don't have like a good front door to that, where here's the data and here you go, and it's not well described. But I'm going to get to that in a minute. But we do have data sets. We have the EPIC data set, which is a data set of there's a movie. There also the EPIC data set is where you have these images that are sort of fluorescence images that define the centroids of the cells. We have some other biological data sets available as well. Those are just image files. So, we need to get I need to go through and actually make like kind of a front door to the data sets. But I think that's what you may have been asking about. What kind of data sets can we work with? So we have data sets. I just need to make them more accessible. But anyways, and then DevoGraph, of course, is the this is the pipeline for the original set of contributions. Those were from Jiahong Li, Longhui Jiang, and I think Wataru Kawakami. Yeah, because they worked on it the first year. And so they worked on this basic pipeline, and that's in stage 1, stage 2, and stage 3. That's just simply like graphs, not hypergraphs, not temporal graphs, not growing graphs, just regular graphs and graph neural network approaches to graphs. So it's like where you just have the nodes in the edges. And then I think I don't know what miscellaneous is. Yeah, so this is just kind of things from, I think, 2022 that needs to be cleaned up. And then we just have our README and maybe some requirements for running, I think, DevoGraph. So, I guess my what I would tell you is we'd have to first talk about what data sets you need, and then we can move from there into like what kind of area do we want to contribute to? Do we want to do the regular graphs or the hypergraphs or the growing graphs? And we're working on. I know we already have Lalith working on the hypergraphs and Jaya working on the growing graphs, but we always could use more kind of insights into that. I guess one area I think we haven't really touched in a while are the regular flavored graph neural networks and the topological data analysis there. I know Paki did some work with topological data analysis of the growing neural networks last year. So there's this package that it was let me see was Himanshu and then Paki, and they worked from this package, which was the it's in here maybe. Yeah, it's this. So NCA is the package that they use for growing the graph neural networks, and then there's this topological data analysis package. And I'm not putting my finger on it now, but like basically it's a way to sort of render the topological features in the graph. And that's something that, again, the topological stuff, we've taken a couple of slippery grabs at it, but we haven't gotten it perfected in any way. So, there are a lot of kind of open areas that we can talk about. If any of those sound interesting to you, we can kind of talk about what kind of specific data we need for that, and then moving forward, how you might contribute.
Thank you so much for your explanation. So basically, I'll check out the dataset and I'll see which part interests me the most. Right now, I have gone through the growing CNN and the hypergraph representation, but I will also look through some of the open sections which are not being touched, and based on it, we will discuss in next meeting. And talking about me, basically, one thing is basically I learned about GNN from Stanford online from YouTube. So there I learned about some kind of graph generation, like generative graph neural network. So that fascinated me. So earlier I thought here we could implement something like that, generative graph neural network, so that we could predict the future growing of silicon or something. So in that, if I get something in that section, that direction, then I could start working.
Okay, that sounds good. Yeah, look forward to hearing more. And again, contact, let's get in touch if you have specific questions. Okay. Let's see. We had some comments in the chat here. So this was, that was Vidhi had talked about or had congratulated Mehul about working with AI agents, and then Lalith, Vidhi, did Himanshu work on 2 projects? Yes, he did actually. He was, and you can do this in GSoC. He applied to the Orthogonal Lab for Open Source Sustainability in 2022. And then he worked on DevoGraph in 2023. So yeah. And then Mehul says right now I'm more into developing the product. There you can see applications. This is for the AI agents. Right now it's in the preliminary phase. Would love to share more details in the future. Yeah, that'd be good. Okay, let me go over one more thing here. So this is for the DevoWorm group. So this isn't just DevoGraph, but this is like DevoWorm. And this is our presence on Huggingface. So if you're familiar with Huggingface, it's this open source repository for data sets, for models, AI models, Um, and people share AI models. There are all sorts of large language models and machine learning models and all sorts of things hosted on this space or the site. And you might have your organization on there or a personal site, and people post their models and they share them. People download them. We haven't really kept this up, but this was actually let's see, this was Sushmanth who did, who was also a GSoC student, I think in 2023. And he did a lot of work on putting this together and putting the models up from DevoLearn. So DevoLearn, the models are up here. So this is our presence, Huggingface.co DevoWorm group. And we have this, we haven't used this in a while, but it's just something I'd like to get in better shape, particularly with respect to the DevoGraph models. So maybe we'll bring this up later and talk about how we can, when we start doing some things for the summer, we can start committing things here as well as GitHub. And so we have our presence here. Actually, we have a lot of repositories on GitHub, and it lists our repositories here. So if you're interested in looking through the repositories, and I know that people are busy with things, but this is just kind of things that we've done in the past in DevoWorm. And I don't know if people are listening out there are interested in some of these repositories, but if you're interested, let us know. Some of these have to do with, say, like the developmental connectome. Some of these we have, I think, DevoWorm, which is where we have a lot of data. Yeah, this one here, DevoWorm. This is where we have a lot of the secondary data hosted for the embryo. And we have some other things as well. We've done some things with CompuCell 3D and some other programs that we've developed. And then this is an important thing here where we have these spaces. So the spaces are where we host our models. So if you're interested in segmentation of images, we have software for that. Sushmanth worked on, so this started as a basic pre-trained model back in 2019. So we wanted to have a pre-trained model for embryogenesis data, for microscopy data, it characterized embryogenesis. And then that grew into some more sophisticated software where we, implemented things like GANs that would simulate embryos and things like that. And then, an analysis of the lineage tree. And then we ended up with, Sushmanth was interested in the segment anything models that it came out of Meta. And so he implemented some of those in the packages. So basically what we have from DevoLearn are these three packages here. We have the nucleus segmenter, lineage population, and membrane segmenter. So if you take an image, a microscopy image of an embryo, it's usually like a bunch of cells on a slide in this clump. And you'll have like cells that are dividing, so their nuclei split apart, and then the membrane, there's a ring that closes off the membrane so that you get two cells, two membranes that become separate with each containing organelles, and then of course a nucleus. And so when you're doing image analysis of embryos, you need to be able to segment out the nuclei, which should be visible in, say, what they call bright field images, or sometimes in fluorescent images, there's usually like a green or a red marker that denotes the nucleus. And so we can segment out nuclei, and that defines usually, sometimes it's a cell, sometimes it's just the nucleus, but we're interested in the nucleus as sort of a unit in the embryo. And then we can, from there, once we segment our nuclei, we can build networks. There's also the membrane segmenter, which allows you to segment the outer edge of the cell or the membrane, and that can be marked with some fluorescent dye as well. Or you can sometimes get them out of bright field images or regular, just kind of black and white images, if the resolution is good enough. And this is a segmenter that allows you to segment out that feature. So we have tools to segment out features of embryos. If you run across the data set where it's like, microscopy data, we have tools for this. And then the lineage population model, which is where, of course, in embryos, you have cells that divide, and they divide according to this lineage. And in C. elegans, the lineage is very specified. So we have the ability to sort of calculate out that lineage tree, calculate out the population of cells in each sublineage, and then do some other things to it. Basically, an analysis on that. I didn't know why this says runtime error, but basically these models, they were, I think they're probably, they haven't been really updated in about 2 years. So, but I think they still work. I don't think that's a problem. I think it's just a matter of, making sure that they're state of the art. But anyways, so we have those three, and then the idea would be that we'd have a fourth and maybe a fifth and maybe a sixth that have to do with the graph neural networks, or maybe like some topological data. So we go from like imaging, go from cell images to some sort of machine learning to some sort of characterization of the data, and then to these graphs and higher level topological analysis. So, and then of course we have in the Huggingface, we have data sets which need to be, this area needs to be updated. This will probably be something I'll be involved with. Just getting the data sets like consistent, so we have like people can come here, oh yeah, these are data sets that are available, these are the models, and then they can do something right from there. So that's kind of a vision for Huggingface space there. It's fallen into somewhat of disrepair, but that's what happens in open source projects. They don't always, anyways.
Hello all. Am I audible now?
Mm-hmm.
Okay, great. So I just wanted to give a quick ground update about what I've done this week.
Okay.
So I'll first quickly share the code. So later I'll let know what the details have been added into it. So the first is I have created a data loader where this takes the features of the mother cell, the XYZ coordinates, the cell, and the time event it happened. So in order to find out and model the growing of a hypergraph at each time step, so we need the time, the actual mother cell, and how the cell was divided, like into the daughter cells, and the coordinates where the cell division was happened, the fission was happened. So for that, what I have considered is this full, the part of the code does is it captures the features. So what are the three distinguished features are? So the first is it captures the lineage depth, because we cannot directly capture the, I have seen this nice, the representation of the lineage tree, how it was developed. So, and based on this, I thought, okay, lineage depth information could be useful in order to model the, while finding out a good relationship in acquiring certain set of, cells coming into a same hypergraph in a hypernode. So what I have considered is I consider lineage depth, the XYZ coordinates as a node features, and then what we get is basically the lineage edges. So in a hyper edge, so we have to model in a simple dyadic edge. We have a an edge between two single nodes, which means that okay, there is a connection between point A to point B. But whereas in hypergraph or in the hypernodes, we have connection, not just a dyadic connection, but it can connect a certain set of nodes. So that's a hypernode. In that hypernode, what we have included is lineage edges. So here the edge information which I have given here, the lineage edges. So we are able to map the two key features. So one is like the time points where the which time point it has been acquired, the node features, and also the lineage node features, both the lineage edges and also the spatial edges. So we'll be able to see these are all the features that we actually capture. So with this, we're gonna see that I have written data letter quickly visualizing what it extracted at a given time points. So this was the C growing hypergraph data loader where it captures the. I have given like max cells per for a given time point and maximum time points 3 and the spatial radius, the radius from which they all belong to the same hypergraph, like hypernode is like 15 I have put. So doing that, I was able to get the results like this. So if this is the mother cell, so nearest to 15 in a certain cells, we are able to acquire the the hypernode features. And this is for the each time point. This is cumulative time point 1. This is the cumulative time point 2. This is the cumulative time point 3. So, and it grows. This is just an example how it grows. When I also tried visualizing 2D, but it was not very intuitive, and it's like overlapping on top of cells, and we are unable to get a clear estimate of where it has been divided. So a 3D visualization, which I've done previously, I included that into this whole framework. So I have a few questions from my side, and I wanted to discuss that. So while doing this, so my question was, okay, I have acquired features of like time points, node features, and all. But my basic question is, if I am trying to model, so suppose this is a cell, and these are all the like this is the cell divided, and these are the parent and daughter cells. And now, if I'm trying to design a hypergraph which should model the total embryo genetic development, so during that process I need to understand I need to understand to model that, okay, how are we trying to how are we modeling a given hypergraph to say that, okay, now we have learned the time point 1, saying that these set of cells, we brought them together into a hypergraph, and in the next time point, and they slowly evolve. So as we see here, the number of cells here starting from the parent cell have been comparatively increased. So the green are the newer cells and blue are the like next time point newer cells. So here we are able to see that okay, from here this one time point we are able to see all these cells. And when we go around like the next time point, the number of cells have been grown, and here for the blue and much more they have been grown. So this is like a total developmental process. So what we need to understand here is what kind of cells we are trying to bring together, the first question. So like, should we model in a way that I'm trying to model in a way anyway my idea was to include both the lineage information as well as the spatial location information. So here when I include the lineage information, so here it starts with this and how this cell divided, and from this what kind of further connections it has. Like here we have this as a parent hypergraph, so which I haven't labeled properly, but this is I think the first cell, which is P0, and from which A B it divides generally, and from AB the tree actually grows. So it's in this certain fashion. So and while division, my question was to understand, okay, we are trying to bring them together. So what kind of, at each and every step, what do we need to consider in order to bring certain set of cells together? So that is the first question. Okay. And the second is, and why we are only trying to understand what kind of cells come together during the developmental process. So during the developmental process, in each and every time step, the model has to learn that, okay, the cell starts from this and then it evolves into this. So that's how it generally evolves. While during the evolution, so it has to understand how it does the fission from a certain cell to another two cells. So where the fission has started and where it has ended. So we have the 3D time like 3-dimensional time points, and with that we also have the lineage information. Okay, from where it started, how it grows. So I'm trying to currently understand, okay, which of them we have to group. That is the first step. The question is, so we are trying to design this hypergraph neural network just to understand the developmental process. And during this developmental process, our hypergraph should give us the information of which cells are coming together. So, I think it's kind of a looped question, but maybe some discussions could help me understand.
Yeah. So, let's see. Mehul asked a question in the chat. He said, I missed a few points. Is closeness of the cells in the graph based on lineage or proximity? Or both? I think Lalith mentioned. I missed to hear it.
Okay, so the first when I'm trying to get the nodes together, we have used the proximity information, and based on that proximity information, we also feeded the kind of the lineage depth from where it started and how depth it has been gone to. So for that depth information, so this is the kind of tree. So suppose I say, while calculating the depth of suppose like this cell, so which could be like, it has been divided into A, B, and then A, R, P, A. Then so this whole depth will be basically the length of this A, B, A, R, P, the 5 times P and 2A. So this could be the depth information from which it has been started. Yeah. So we are also feeding the depth information and the proximity information of XYZ coordinates, and also the 3-dimensional locations, and where I have been returned this thing. So just give me a second. Yeah. So here the time point, the node features, the lineage, and the spatial hyperedges. So this is what we get from the whole data set, and yeah. So what could be a good way of modeling? How do we bring them together? Because when we develop a hypergraph right now, so if we wanted to bring the attention mechanism, when we certainly say that, okay, we are trying to understand that, okay, the nearest 50 cells, they should come together, nearest 5 cells should come together, nearest 10 cells should come together. When I say that, then they come together, and when we apply these hypergraphs and attention mechanisms, the information that is shared among these 10 or 15 or 20 cells will aggregate, and it forms a network which tries to understand what is the relationship among these set of the grouped set of cells. So maybe when we are trying to understand syncytiums, we say that epithelial cells, the motor neurons, these neurons, okay, they form a certain pattern, right? So when they come together, so the information is shared across the syncytium. So when we are modeling this from time point 0 to like time point n, so during this developmental process, we need to understand which cells go together. For that, initially, how are we starting is my question. How do we have to start?
Okay, so I think I can answer your question like this. So you showed the lineage tree, and the lineage tree is organized, of course, it has some depth. It's actually organized by sort of anterior to posterior, and so the left-hand side is like towards the front of the worm, the left-hand side, the back, or the right side is towards the back of the worm. And so that's expressed, of course, in the coordinates. So the coordinates should basically reflect that, although there's some migration of cells. So, right. But then you want the categories. So the categories themselves, we have that information as well. we have the sublineage, and in the C. elegans lineage tree, we have 8 sublineages. We have there are 4 in the AB lineage, and then 4 in the P1 lineage. And there's a list. If you look up C. elegans sublineages, there's a definition of them. So we can classify them like that. We can just say, like there's a cell that divides, and then that cell has an identity. And then it has a sublineage that it belongs to. So that has a label of a sublineage.
Right.
And then you can have those, and those sublineages usually have some thing that they're going to differentiate into. So if you go to the lineage tree and look at and this is well known in C. elegans, that's why I'm suggesting it. Each sublineage will differentiate into certain types of tissues. And so like the stem cells, or there will be germ cells that'll be restricted to germ cells. There will be like in, I think, sublineage C, it's it's a certain cell type. In MS, it's a certain cell type. And then in the AB sublineages are a little bit more diverse in terms of what they'll turn into. So we could label cells with a sort of potentiation where they could become a number of things. And then we can also go to the once in if you get deep enough in the lineage tree, we start to get differentiated cells. And so at some point we're going to get differentiated cells, and those definitely have an identity. So those are neurons or epithelial cells or muscle cells. And so when you get to that point, then you can label those with a specific label. So it's kind of coming from there's an identity of being part of a sublineage. There's an identity of what that cell might turn into. And then there's this identity of a terminal division where you don't get any more divisions, but those cells exist in the tree that definitely have a category that they should be in. And so that's kind of how I would approach it. And all that information is available if you go to the interactive C. elegans lineage tree, which is online. That's all laid out. You can go explore the tree or get information for different sublineages. There's also a lot of information from the C. elegans community on sublineages, and there are 8 sublineages, and then those sublineages they don't all become just anything. Some of them, there's a certain number of set of fates that some of these sublineages will turn into. So that's what I would say. We can use labels for some of these potential fates or some of the nomenclature labels, and see that we can use those to group them into hyper nodes. And then I don't know, it'll be a little bit more complicated than that because there's probably going to be some ambiguity, but with C. elegans usually there isn't a lot of ambiguity compared to other organisms.
So, sorry, you have mentioned that the fate information, in the sense the cell fate. So it's kind of will it survive or not? Is the
yeah. So in C. elegans, these cells divide, and they're on the way to becoming something. Some of them die before they're differentiated, but basically, this information I can pull it up for you too. There's basically a map where you can say this sublineage will become maybe epithelial cells or muscle cells. This sublineage will become all muscle cells. So you have a number of different possibilities depending on what specific cell you're talking about at what level. But then they always have this label that's like a sublineage. So you can use the sublineage label. You can use the potential, the potential what cell that sublineage might turn into, or you could use just a spatial criteria where these cells are in the anterior part, these cells are in the posterior part, and then just have different hyper nodes based on that. I'm guessing that you're going to end up with a lot of different possible hypergraphs from this.
Exactly.
Yeah.
Yeah.
So you just
So
have to kind of think about what the best criterion would be.
right.
Yeah.
And also, in order to learn this, the temporal relationship. I mean, like at each time point, when we develop a hypergraph which has certain set of connections, so it grows over the time. In order this neural network to understand the whole, that okay, at time step t, these are the cells, and at time step t plus one, these are the cells that newly added from these set of parent to child. So this expansion, what happens? So is of my interest, and when we are trying to model that in certain way, we have to make the neural network. The hypergraph neural network make it learn in such a way that at each time point it evolves in this certain fashion. So in order to make it learn, we have to find out a certain objective function. So suppose we are saying that when we are trying to do a simple classification task, what we generally do is we say that okay, these group when we are trying to do node classification on certain set of tasks, we classify each and every node saying that okay, these cells belong to this, and or this group belong to this, and based on that we do classification. But when it comes to understanding the developmental process, so it's more like the hypergraph itself has to acquire the information of the whole the whole developmental process. So how the neurons, the neural developmental process in the sense, so how these neurons have been established after these set of time points of cell division during this embryogenesis, how they have been developed. So this, in order to understand, there needs to be a certain way of training the neural network. Maybe in the next week I'll read up something, maybe, and I will discuss some of the criteria in which we can model it, and maybe I'll read about the literature in this sublineage tree information, and also I'll look into some of the results and bring some good updates.
Okay. Yeah, that's great. So yeah, then a word about I guess what you're saying is it needs to maybe learn the adult phenotype or where things end up in the adult.
Right.
To learn, yeah. But the thing is, we don't have a similar model of the adult. We don't. I mean, we have you've seen the Blender model that OpenWorm has, but those aren't I don't know if those fit into a coordinate system that is equivalent to what we have for development. But we can. I mean, I guess we could train it on basic information about the sort of the cell lineage, say this cell is at this, like I said, the cell lineage tree is organized anterior to posterior, left to right. So if you just take each cell's position in that tree, how far is it from the far left-hand side, and then working your way right. We actually did this years ago in a paper where we did a binary decomposition of the tree.
Oh.
And then basically came up with binary values for every cell in the lineage tree.
Oh.
It's just a matter of the far left hand side would be zeros and the far right hand side would be ones, and or it's like a really long vector, but it would just be every division would be its own the next higher binary values. You work your way right. And I don't know if that needs to happen, but that would be kind of an addressable system for the lineage tree. I mean, it would be kind of hard because it's a lot of work, but
yeah. So, but it's very interesting. So that could actually help if when we are trying to calculate depth information or something sort. So I think that will definitely help, because the numerical systems we can directly feed to the machine, so that we can label them based on the information that we have. And I think that that actually very interesting and useful. Yeah.
Okay. Well, yeah. Keep working on that. That looks great.
Thank you.
Make sure you push some of that up to the
sure.
GitHub. Yeah.
I'll do this. Saturday. Yeah.
Okay, that's fine. Yeah. All right. So I wanted. Oh, I don't know if Morgan had anything to say.
No, I mean everything's looking great. It's coming along. I mean, the only thing I can report from the Neurotech summits. So sorry, I dumped a bunch of notes into network neuroscience, but I will follow up on those on Saturday.
Okay.
And but Conrad Cording was there, and I asked about this and was not pitching a C. elegans institute with Boyden at this meeting. Yeah, there is no institute yet. They are still looking for funding, and yeah. So I forget where we saw that original post, but like I think that was just foresight trying to imagine a wonderful future, and so it's super interesting. I mean, I love all the progress that people are making. I hadn't seen the Huggingface DevoWorm group before, so I'll definitely try to look at that, and maybe I can like add some of the data sets that you want listed there, things like that.
Yeah.
So that's super cool. And this weekend, it's not directly relevant, but this weekend is the Huggingface Le Robot Hackathon, global hackathon. And here at the tower in San Francisco, we are one of the official nodes. So people are putting together robot arms for that hackathon, and it should be pretty cool. We're doing a biopunk hackathon at the same time. I have not yet figured out a way to link in DevoWorm into that, but would love to have some project eventually that actually is maybe contributing data or something.
Yeah, that's great. Thank you. Yeah, and looking forward, to hearing more about the hackathon and some of the things going on there, and
yeah, and then I've sort of, or I'll definitely be asking about things, but next week I'll be at the Allen Institute for this open science for undergraduate education.
Okay.
And I don't know how much will be kind of DevoWorm specifically relevant, but I will again be keeping my eyes out. Yeah, I'll post links for the hackathon for sure. And yeah, so but definitely we'll be learning about like the Allen Cell Atlas and things like that are developments that we're doing relate to the Open eFiz platform. So yeah.
Yeah, that's great. All right. One last thing I want to talk about today. Mehul, you had something to say?
Yeah, sorry. Like my question was like on the Lalith's part. I missed to ask that time. So like I think Bradley like summarized things. Asked like whether like my understanding is clear. So like we can categorize the cell into like various types, like you mentioned, some like types of tissues, whether it's like stem cells, germ cells, or maybe some other subtype, like AB subtype or A and P. And then there are like cells type, right? Epithelial, you mentioned muscle cells. So all these can be the information to provide to do a certain type of task, right? So where Lalith was mentioning the objective. So here I was confused like what task we can do. So one task like mentioned was like binary decomposition, like far left or far right. So here we can figure out the objective function. I think that would be straightforward. Like we can try with like basic ones, starting with distance metric. Yeah, so I was thinking like what tasks we can do. So can we also do like so one task is like this decomposition in terms of like coordinates or the proximity, right? Can we also do tasks like, let's say we have the training feature of coordinates, like we provide a feature in coordinates, and then we do the task of self rate prediction, like which you said you'll be providing data also. So I'm just thinking like what all different types of data we can feed and what different tasks we can do. So yeah.
There are a lot of possibilities. Although the thing about C. elegans is that those things are already known. Like the self fates are pretty well known. It's just that when you're dealing at the level of, if you're at the top of a lineage tree, you know this stuff, so you can train the model. But then you also have things that are kind of like you're starting here and you're working your way down, and the process, it's almost you can do a very good job of predicting the process because you already know what it's going to look like at the end. There isn't like it isn't a lot. There isn't a lot of like generative aspect to it, if you know what I mean. Like it doesn't change, or there aren't different combinations as much as you would get in say like a mouse embryo or something like that. So kind of what the end state is more or less. So you can train it with that, and then you can like let the process run, and it'll just kind of predict itself. We did some work several years ago on like predicting lineages, and this is in the DevoLearn repository, where we predicted different lineages based on the data that was already known in the lineage tree, and just generating like I don't remember how it was done, but generating a lineage in from images and generating them from counting the cells and then predicting against the lineage tree. So it's like taking data that's been collected, analyzing it, and then fitting it to the lineage tree. And you could do the same thing here, where you just generate a process where you're generating like a binary tree, and then predicting the lineage tree or something like that. I'm not gonna sit here and tell you that I have that all worked out in my head, but that would be the kind of thing that we could do. And there's a lot of stuff in like the DevoLearn repository that really I think maybe can be brought to bear on this, as well as I'll have to bring up the binary decomposition work we did, because to a certain point, we do have like the binary values for each cell in the lineage tree. I think up to the 256 cell stage. So that's, but it's like and that's just another version of a coordinate system. And we already have positions, but then we would have that. All right. Yeah.
Makes sense. Sorry, I also had like one question for Lalith. I would see his code like when he puts it on GitHub. So he mentioned like there are like features like parent, proximity, time. I think there was like one more I'm missing. So how, Lalith, like how are you encoding these features? Like, sorry, I'm going to very basics. I didn't understand it like properly. Are you encoding it as like graph features, node features, head features? Like how did you plan to do it? Like time, proximity, and lineage. Yeah.
Right. So maybe I'll show. So we cannot directly give the lineage information, that is like P0, P1, P2, I mean like the tree information, right? So what I have tried is I gave the the depth of the tree information. So when I say lineage depth, so from where it started and from where it ended. So suppose AB divided into ABP and AB, I'm not really sure, but ABC, suppose. So, and how far this has been gone in the given proximity time. So what is this proximity is? We're gonna group, I fix a certain radius, say 15. So from that cell point division, I will calculate that whether there are like, I will see how close 15 are, like similar to that of KNN. So 15, how 15 cells are, by near proximity. From that 15 cells, we take the information of XYZ and also the lineage depth. So these are the three information that I code. So in order to calculate the distance, a simple Euclidean distance I follow for the, while doing this, the proximity. And when we feed that information, we give, node features. These are like each and every node carries these features. These node features are nothing but the lineage depth, the x, y, z coordinates. And for the edges, I give both the lineage edges information and also spatial hyper edge information. Spatial hyper edge in the sense which boundary it actually captures. So this hyper edge information comes from the KNN part. So from which boundary that I have drawn. So that's one spatial hyper edge. And lineage edges are the edges which have been captured from from the parent to the child, how far it has been gone to. That we append the information. So this information we give it to the hypergraph. So we say this is an hypergraph. So the proximity is basically the closest. This is a parameter that we can fix. That is, suppose say we can say 5, 10. So it differs. And so for visualizations, I put the max time point so that it's quite easy to do that. So this we can put it max. So did I?
Yeah. Thanks for explaining.
Is it
it makes sense.
Enough? So, but because you have already.
So I actually don't have a lot of context. I did it long back, but I'm still very curious. So you are making a big hypergraph, which is starting from T1 to T0 to the last T point, not like a temporal fashion, like a complete single big hypergraph, which has time also as a single feature. And you are putting a threshold at T equal 3 in this particular case.
Yes.
Okay, so time was the feature, and distance proximity was a feature, and third was a lineage feature, just to make the hyper edges. Got it. Okay. Thanks. Noted. Thanks for the information.
All right. Yeah, thank you. So let me go one more thing before we go here. Wanted to bring people's attention to this paper. This is for Vidhi's benefit, but we also have other people working on this. This is the open source community sustainability project where we have this white paper or preprint where we've been trying to update this to reflect the progress in the project. And so this paper reflects what was done in 2022 and 2023, but not 2024 and then this year. So this is open source community sustainability using agent-based models. And we have Himanshu and Brian on it, as well as myself and Jesse. And we're trying to update this. We've been trying to update this. So it probably needs to be updated with a couple other authors and some more information. So the paper basically walks through kind of the project, what our goals are, the kinds of approaches we've taken. So this kind of motivates the problem. And then we have different models for collective behavior. So we have cybernetic big five theory, reinforcement learning, collective active intelligence, and collective active intelligence is a active inference approach that was a Brian McCorkle. Cybernetic big five theory actually was Hussein Ather, and I think he's also an author on this. It didn't show up on the front page, but I think it's on the paper. And then reinforcement learning was Himanshu in I think 2023, and then we've done some more reinforcement learning work, in 2024 or in 2024, and then we did some large language model work last year with Sarah, and then this year with Vidhi. So I want to add in the large language model stuff, integrating large language models with agent-based models, updating and reinforcement learning stuff, and then that would be the next version of this paper. So we actually have a table here. It shows the different models and the different parameters that have been used. This is just a matter of organizing things that are in that repository and presenting it as a unified project. So there are all sorts of different things in here from the different projects. And so what I'd like to do at some point here, and this has been a long time coming, or a long time suffering, I guess, is to update this paper according to some of the things that we've learned since this paper was put out. I think this was put out in 2023, so it needs to be updated. And we learned a lot last year. Actually, it was very, very productive summer. But that's what I want to do with this paper. So we have a couple of chat. We have a couple things in the chat. So this is VDS. Can you drop the link in the chat if possible? Yeah, I'll drop it in the Slack. I'll put the paper in the Slack and you can go over it. Yeah. Okay, any more questions or comments before we finish up for today?
Yeah, I do have a question. It's
Okay.
not really related to what we are doing. I can ask tomorrow as well, like we can discuss this tomorrow. So, actually, in my university, we have access to the virtual reality headsets. So we wanted to go ahead and like me and a few of my friends, we wanted to go ahead and actually learn virtual reality and how it works, but we don't know how to go ahead. So do you have any inputs on like how we should start or something? Like for now, all we thought is that we have the headsets, so we can somehow create the virtual environment. Like it can be some underwater environment or something, or like a very small environment because we're just starting. So we don't know how to start. Like we just have the idea in our mind, but we don't know how to start. So like I obviously start this after the GSoC period ends, so probably towards the end. But we're just thinking about how we can go ahead. So
yeah, so there I guess there are all sorts of you're talking about programming an environment for that.
yeah, like anything in virtual reality, like we want to use the headsets, but we don't know like how to start or anything. So do you have any prior experience in it?
What headsets are they?
I think we have MetaQuest and Apple Vision. So
Okay, so yeah, if there are developer tools for each headset, so it's kind of hard to say this is what you should do. Like with something like the Quest, there are tools available. People have, if you go to GitHub, people have developed simple environments, things like that. They're usually people come up with very simple demos. I know Hussein, who's not in the meeting here, but he was working on a simulation of something called the Moritz Water Maze. I'm not sure what this. And then we also had some other person, another student who was working on implementing some tools in VR. And I think we have that repository in the GitHub, in the organizational GitHub. So if there's a VR DR repository in our GitHub organization for Orthogonal Lab, and there are things there that were sort of the basis for some development. So those are some resources to check out. There are also some other you can probably, with Quest, it's easy because you have like developer tools and you have like things that people have worked on. And I would just start very simply. I wouldn't go overboard. There are also very simple games that you can use on the Quest that are, just demos. Just like I want to show how this works. So it really depends on what you want to do. If you want to just play with it to see what you can do in it, or if you want to develop for it, which is a little bit higher bar.
so like does the code base like we have to connect the MetaQuest with some code base, right? And that will be in Python itself.
Yeah,
How does
yeah.
it work? Like how do we create the environment? Is it like it's like how we use Python libraries like Pygame and stuff, and then we create games.
Yeah.
Is it something like that?
Yeah, I'm not, well, you can use like Blender. So you can use that. You can model 3D objects and you can put them in a framework. You can use, I think maybe Pygame has some tools, or there are other toolboxes. But yeah, I don't know what the standard is for like developing directly for the MetaQuest. There might be tools that make it very easy to do like very simple demos. I'm not familiar with what the name of those tools are.
I will go ahead and check the AR/VR repository, and also in tomorrow's beautiful meeting also I'll ask just in case Jesse and Morgan have some input.
Yeah, it'll be good. Yeah.
Thanks.
All right. Yeah, and Mehul said in his and Lalith's lab we had some Oculus, if I'm not wrong. Yeah, they're very common, so the tools are out there for the Quest. It's the easiest one probably to develop demos for. Yeah, cockside experiments. Yeah, then definitely. All right. Well, thanks for attending, and good luck for the next week of work. Thank you.